All right. So today will be your experiment number 6. Okay. DNA and protein sequencing. Okay. Page 24. All right. So kita buka muka surat tu sekarang. Okay. All right. So there will be two activity in the experiment. Okay. Number one will be analyzing DNA sequence using APE software. Okay. Another one will be activity number 2. Okay. All right. So introduction about this experiment. All right. Number one, you have to know what is APE software. It's a small program for displaying, analyzing DNA sequence. Okay. It's a free software. Okay, which is you can download it on this. Okay. On this website. All right. So please make sure you have a latest one of the APE software, right? To access the APE software, okay. Make sure latest one, all right. So why we need this APE software? It's a basic feature to find useful information about particular DNA sequence. Okay. So you can take is it the free software? No need for you to online. Okay. It's very simple. You just take and then do the software in the following all the steps. All right. It's a very simple software. Okay. Okay. Contribution to the industrial, okay, use and commercial knowledge. DNA sequencing is the process to determine the precise order nucleotide within DNA molecules. All right. It includes any methods of technology for what to use to determine the 4 bases, which is adenine, guanine, cytosine, and thymine, which is the strands of DNA. Okay. So the technology was to imagine the desirable character can be improved in the living body by controlling the expression of the target genes. Okay. So we use this technology for what? For crucial protein requires for health problem, dietary purpose, can be produced safely, affordably, and sufficiently, right? So this technology, multidiscipline application and potential to deal with the important aspects of life, all right? Number 2, improving health, enhancing food resource, resistant divergent of adverse environment effects. Okay. All right. Please pay attention to do all the exercises. You will need this for your test. All right. Okay. What is the main objective for this experiment? Okay. Number 1, to analyze DNA sequence using computer. Right. Okay, there's several methods that we use in this experiment. Number 1, you must have a very good computer with the latest Windows operating system, OS, or laptop, right? Number 2, you must have a APE software. Make sure it's the latest version. So just download it on the website above. Yang tadi tu, alright? And then you must open the NCBI website. Why? You need to download a FASTA sequence there. Okay. All right, I will show you the process how you download the APE software, okay? And then you get the FASTA sequence from NCBI, okay? Alright, first of all, you have to open your window, your internet, okay, Google's, Chrome's, and so on and so forth, and then type APE software, right? Okay, and then you go to here, okay, Jorgensen Biology Utah Edu, alright, and then click on APE, a plasmid editor, right? And then you will get this interface, okay. There are two operating system that you can download, okay. Number one will be OS or in Apple computer. Number two will be Windows, right? So make sure you know this is the latest one, nampak ni, the latest one APE software with the version 2.061 in February 5th, 2020, right? Kalau awak punya operating system komputer awak adalah Apple, awak pakai OS ni, okay. Kalau awak punya komputer biasa pakai Windows, awak ambil download dekat sini. Alright. So the rest words kat bawah ni adalah apa tu, soal jawab, okay. And what you can get from here, alright. And then that's all, right? Okay, and then you have to click on the which OS you use in your computer. So kalau macam komputer saya pakai Windows. Click on the window, right? And then there will be downloading the APA Win current in zip. Okay. So apa perlu buat? Awak buka dekat mana awak punya APA Win current zip ni, lepas tu awak buka. Alright, buka zip ni, boleh. Okay. Bila awak dah buka ni, alright, awak akan dapat apa Okay, awak akan dapat interface ni. Alright. Interface like this, okay. Inilah dia software yang kita nak, APE software tersebut. Alright. Kalau awak tengok software dia macam mana, dia ada files, okay. Awak boleh klik files, edit, enzymes, OFS, features, tools, window and helps. Okay. Bila awak turunkan awak punya cursor, akan ada, awak klik pada ikon tu, dia akan beritahu nama dia. Apa ikon-ikon tersebut So this is your interface for APE software. Right? So gunakan ini untuk kita punya eksperimen hari ini. Right? Okay. Alright, so sekarang ni saya akan tunjukkan macam mana cara awak nak dapatkan data daripada NCBI pula. Okay. Okay, macam biasa open your Google, alright, and then click type NCBI. Okay, so sekarang ni awak akan nampak National Center for Biotechnology Informations. Klik kat situ So you get this interface alright. You will get this interface. Okay, bila dah dapat ni awak kena klik dekat all database tu. Okay. Klik kat all database ni dan cari perkataan nucleotide. Okay. So bila dah dapat nucleotide tersebut, awak type lah spesies apa yang kita akan pakai hari ni. So hari ni kita akan pakai Lambda strains. Okay. Right. Dan klik search. Okay, dia akan keluarkan banyak ni. Alright, nampak ni. Dia akan keluar banyak ni. So pilihlah salah satu. So kita pakai Bacillus cereus N7401. Okay, awak klik pada sini. Okay, so sebelum tu bila awak tengok apa tu interface tadi, okay, so dekat atas ni sebagai nama this one is the name for the genes, okay, this one is represent the size of our base pair or DNA sequencer, okay. So dekat sini dah dapat gene bank ada, FASTA pun ada. So sekarang saya akan ajar awak macam mana kita nak download FASTA tersebut, alright. Klik kat nama ni. So akan keluar benda ni, alright. Okay. So bila awak scroll bawah, awak akan nampak ada sekuen-sekuen dia, alright. Okay. So and then macam mana kita nak download benda ni, kita tekan FASTA. Okay. So bila awak tekan FASTA, dia akan keluarkan kita punya all the nucleotide inside this species, alright, Bacillus cereus. Okay. So that's meaning of nucleotide okay. So macam mana awak nak downloadkan awak punya apa tu FASTA ni, data ni, okay. Quickly you click send to. Alright. Complete records, right? Choose destination, you put file. Click on file and then the format will be FASTA and then create file. Bila tekan file ni, dia akan downloadkan untuk awak. Nampak tak bawah tu? Dia akan jadi sequence tu alright. Sequence FASTA akan masuk ke dalam awak punya download icon dalam awak punya komputer, alright. So awak buka ni. Okay, so awak buka sequence FASTA ni, awak renamekan balik dengan nama Bacillus cereus tadi. Okay, so sekarang awak dah ada awak punya FASTA sequence untuk meneruskan practical ni. Right? Okay, continue. Okay, now you have what we call APE software, awak dah download, and then you have already download the FASTA sequence from NCBI websites. Okay, and then we go for the methods. Alright. So you have to double click your APE software first to launch the program. Okay. So number 2, a series of icons. Okay, kalau buka tadi ada nampak gambar-gambar tadi alright. Will be available in the toolbars. Okay, write down the function of each icon from A to R. So A to R apa dia? Icon dia. Icon tu adalah gambar-gambar ni. Okay. So saya beritahu tadi macam ni kita nak tahu gambar-gambar tersebut, awak kena klik setiap gambar icon tersebut, dia akan keluarkan nama. Alright. So awak kena tulis A sampai R. Apa dia? Okay. So this is the interface of our our APE software ataupun nama lain adalah editor sequence editor window Okay, we go for number 3. Please open the file on the icon of toolbar. Okay, and then selected the folder APE files from the desktop yang kita dapat tadi. Alright, so macam mana cara dia? Awak kena open file, lepas tu ambil file yang awak guna tadi. Okay, alright. Okay, so kita buka balik kita punya window tadi, alright, window this one, window tu. Nanti saya akan tunjukkan macam mana cara awak nak open file awak alright. Okay, so sekarang awak dah ada interface tadi, alright. So interface ni awak kena buka apa Awak kena buka file ataupun awak klik pada icon ni. Open file, okay. Pilih antara satu awak tekan pada file nama file tersebut ataupun awak tekan pada icon. Sama je konsep dia, okay. So saya tekan open file pada icon So dia akan keluar pelbagai kita punya FASTA. So dalam konsep ni saya dah memang download dalam FASTA saya. So saya boleh pilih apa benda yang saya nak So dalam konsep ni saya pilih untuk musculus, alright. Okay. Alright, bila awak dah open apa yang awak dapat, awak akan dapat structure ni. Window ni penuh dengan kita punya sequence yang kita download tadi. Okay, please note bila awak gerakkan awak punya cursor ni, alright, cursor ni awak akan nampak pergerakan nombor tu atas bawah tu, okay. So awak boleh highlight sequence-sequence awak nak tu, alright. Okay, so this is how you open your APE software ataupun editor window, and then you upload your sequence FASTA, right? Okay. Okay, ada soalan? Is there any question? Okay, kalau tak ada kita boleh continue untuk kita ikut prosedur. Okay, saya nak bagi inform dekat sini. So please awak pilih sequence between $1 juta hingga 50 base pair sahaja. Okay, dan spesies tak kisah spesies apa tapi the best adalah bakteria, right? Okay, so awak boleh pilih semua sequence tapi maximum sequence tu daripada 1 juta hingga 50,000 base pair. Alright, and then species saya syorkan adalah pakai bakteria, jangan pakai human dan sebagainya. Ambil saja bakteria ada. Okay, so boleh saya bagi kelugaran dalam kita punya practical ni, tak semestinya awak pakai Bacillus subtilis ataupun Lambda SP strain ni, right? So boleh guna lain species. Okay, tapi base pair tersebut atau awak lingkungan daripada 1 juta hingga 50,000 base pair, right? Alright, continue. Okay, we go for the number 5. Okay, saya kata file Lambdas Training, okay, taken from NCBI website. So saya dah bagi tahu tadi awak tak perlu ambil Lambdas Training, okay, just pakai strand yang ada lebih kurang 1 juta hingga 50,000 minima, alright, maximum 1 million, minimum 550,000 ataupun 50k base pair, right? And then click open, alright. So the sequence will be loaded onto APE, alright. Okay, quickly go for number 6, down, write down the length of lambda or the species that you choose, right? Or the bacteria you choose, right? And then number 7, click on the graphic map on the icon. So icon tu adalah button-button kecil yang awak nampak tu, okay, gambar-gambar, alright. And map the sequence will be displayed, alright. And then click on the linear buttons. So the icon will be changed to what? Alright. So untuk awak gambarkan result ni, awak just print screenkan gambar tu, alright, dan letakkan dalam awak punya report, alright. And then click the graphic map again. Compare with your C in the 7, then close both window. Alright. And change the icon back to linear by clicking it. Alright. And then number 10, move the cursor over the sequence. Okay, yang tadi saya tunjuk tadi. The number on the top left corner change. What does the number represents? Alright. And then click anywhere in this within the sequence. The third number on the top left corner change. What does this number represents? Okay, so awak pilih mana nucleotide awak ada dalam awak punya apa tu? Awak punya editor, awak punya apa API software tu. Lepas tu awak klik satu, letakkan satu cursor dekat satu bahagian tu. Apa maksud dia? Right? And then what the number on the sides on the sequence boxes for? Okay. And then go for the enzyme selector dialog. Okay, 13 You can do this by either clicking on the enzyme selector icons or select enzyme from the toolbar. Okay. And then select the enzyme selector or by pressing Ctrl E. Okay. So pilih enzyme selector kat situ Okay. So bila awak ada select enzyme selector, what do the names and the number in the bracket represent? So this one adalah selepas awak buka enzyme selector tersebut. Okay. So apa yang awak dapat situ akan ada perkataan what we call hint tree. So cari dalam tu ada perkataan nama hint tree. Okay. And now will be highlighted in red. Maknanya bila awak klik dekat nama tu, bila awak buka procedure 13 ni, benda tu akan jadi warna merah Okay, and then place your cursor over the highlighted text and write down the recognition sequence of the enzyme. Okay, and then select the graphic map. So apa awak dapat Okay, awak dah letak awak dah jalankan prosedur nombor 13, dah pilih, contoh pilih satu adalah hint tree. Okay, dah dapatkan highlight merah, and then open balik awak punya graphic map. Okay. So a map of lambda, DNA and position of hint tree recognition restriction are displayed. Okay, this one awak kena buat tengok ada tak. The number indicate the positive position of the site and draw it here. So cara awak nak draw ataupun awak just buat print screen dan lekatkan gambar tu dekat sini. Okay. Ataupun dalam layan report yang baru alright. And then go back to the API window ataupun awak punya apa tu the API window alright, and select digestion window. A figure will open up. It show with your expect to see of a sample of lambda DNA ataupun spesies yang awak pilih alright, atau bakteria awak pilih. Digest to the completion with the hint tree is ran on the agarose gel. Compare this to your result in experiment number 2. Okay, so untuk this one nombor 17 dengan nombor 17 awak just dapatkan dia punya decision window saja. Tak perlu kita comparekan dengan result eksperimen nombor 2 sebab kita buat eksperimen tersebut right. So just gunakan untuk kepahaman awak apa itu digestion window So once awak tekan digestion window apa yang awak dapat selepas tu please open page 34. Ni gambar raja Write down the each section on figure represent. Nampak ni. Bila awak tekan tekan apa? Tekan digestion window you will get this displayed. Alright. So dalam bentuk macam ni okay. So this one represent the picture yang kita dapat. Apa maksud dia? Maksudnya adalah kita dapat digestion window yang kecil ni. Alright. Okay, 18 review the picture. Okay, this one tak perlu buat Okay, this one hanya untuk awak compare dengan eksperimen number 2, 18 practical number 18. So this one hanya untuk menyatakan bahawa awak faham proses digestion window tu saja Okay. Okay, so this one kalau kita ada result eksperimen number 2, kita kena letak dalam ni dan kita comparekan dengan spesies kita ataupun eksperimen kita. Alright. So procedure number 19, all of you have to do repeat back the exercise except for step number 17 and 18 sebab apa 17 and 18 ni involvement of experiment number 2. So tak perlu buat alright. Tapi awak kena buat eksperimen protokol ataupun procedure number 19 with other enzymes. Okay, for example AF3 then repeat the combination of AF3 and SAC2. Okay, for each exercise you have to do what? You have to write down number 1 A, number of restriction sites. B, position of restriction sites. Okay, so semua ni awak akan tahu bila awak gunakan ikon yang dinamakan sebagai apa? Sebagai enzyme selector, right? C, the number of fragment generated in the digest, okay, D, size of each fragment, okay, and last one, draw a picture of digest. So untuk kita buat benda ni, awak kena print screen sahaja dan letakkan dalam awak punya reports. Right? So untuk eksperimen ni tak perlu buat infographics. Okay. Okay, and then we go for the activity number 2. Okay. What's the meaning of activity number 2? Analyzing protein sequence using APA software. Analyzing protein. So tadi kita buat DNA, now proteins. Okay. So introduction same as the before. Okay. Alright, kita go pada the second part of a lesson of APE. So hopefully you will be familiarized with the basic features. Okay. So this one kita akan belajar tentang apa activity number 2, locating open reading frames, translating DNA sequence into amino acid sequence. Okay. So contribution to the industry use and commercial knowledge, particularly agriculture, the genetic modification plant have augmented resistance to harmful agents, enhance product yield, show increased adaptation for better survivals. Okay. Moreover, recombinant pharmaceuticals are being used confidently and rapid attaining commercial approvals. Okay. So we use what we call technique recombinant DNA technology, gene therapy, and gene modification. Okay. For what? In the purpose of bioremediation and treating serious disease. Okay. For example, compared to conventional breeding, the transfer of large number of both specific and non-specific genes to the recipient, genetic engineering only transfers small blocks of desired genes to the target through various approach, okay, such as biolistic and agrobacterium mediated transform saja. Instead kita tanam pokok yang besar tu, kita cuma just alter DNA dia je, lepas tu kita tanam, alright. So banding proses yang mengambil masa lama, kita just buat dalam masa beberapa minggu ataupun bulan. Yang eksperimen lama beberapa tahun. The alteration into plant genomes is brought either by homologous recombination dependent gene targets or by the nucleus mediated site specific genome modifications. Okay. Recombine mediated site specific genome integrate and organonucleotide directed mutagenics can also be used, right? Itu gunanya kita tahu sequence yang kita nak, right? Okay, the objective for the experiment to analyze DNA sequence using computer, right? So apparatus that we need, alright, computer with the letters OS, right, APE software and NCBI website for downloading the FASTA sequence. Okay. Alright, so methods continue. Launch the APE programs and select open file icon. Okay. For this experiment, okay, saya dah beritahu awak boleh pilih sequence apa awak nak, tak semestinya kita pakai Bacillus cereus ME strains. Okay. Awak boleh pilih sequence tu between 1 million base pair to 50,000 base pair. Okay, the best kita pilih bakteria lah, alright. Okay, so tak perlu pakai Bacillus apa tu? Contoh yang ada dalam kita punya procedure, alright. Okay, number 3, note down the length of the sequence, the start codon and the stop codon, alright. And then find out how many restriction site each of these enzyme in the sequence. Alright, so awak dah tahu eksperimen eksperimen pertama tadi, aktiviti yang pertama. So awak dah tahu okay, awak akan tahu macam mana kita nak tahu berapa banyak restriction site yang kita ada pada EcoR1, APO1, DR1, BHI, BAMHI, HIN3 ataupun RSLI. Okay. So procedure number 5, we digest DNA using PSIL. So this one kita gunakan another species ataupun another sequence. Bukan sequence yang pertama tadi Sequence daripada aktiviti pertama, ini sequence aktiviti untuk kedua, right? So pilih satu je, PSI pula. How many fragment will be produced and what are the size of these fragments? Okay. And then go quickly for the activity number 2, the main activity, locating ORF. Okay. Close all the other window except editor window, the one showing your sequence ataupun kita punya APE software tadi, right? And then tapi APE software tersebut dah dibuka kita punya sequence yang baru, right? Sequence yang kedua, bukan sequence yang baru dan bukan yang aktiviti yang pertama tu, aktiviti kedua. And then you have to select ORF on the menu bar on the top and select the ORF maps. You will see the new window showing the 6 possible open frame. Okay, open reading ataupun open reading frame. So macam mana bentuk dia, awak boleh check dalam gambar rajah yang ni. Okay. So ini adalah gambar rajah dia. Okay. Bila awak buka yang baru, awak tekan ORF, awak buka ORF map, awak akan dapat structure yang ni. Okay. And then goes to the practical number 9. What is reading frame? Okay. Why are these together 6 reading frames? Okay. So number 10, each of the reading frame, a short vertical represent what and a long vertical represent what. Okay, kalau tengok gambar rajah ni ada yang panjang dan ada yang pendek. Kenapa panjang, kenapa pendek? Kena check alright. Okay, we go for the element number 11. Which of the 6 do you think is the correct reading frames and why? Okay. So now close all the window and select ORF again. From the top bar menu, now select the translate. Okay. Select all option as shown below. Okay, so bila awak buka bahagian ikon yang ada nama translate, awak akan dapat macam ni. Okay, so biarkan select pada one letter, alright. Lead win 20, line number none, DNA pun none, and then click OK, alright. So apa awak dapat dari situ? A new window will open up. So macam mana bentuk dia? Dia akan jadi macam ni. Benda ni adalah contoh di mana kita ada 3 nucleotide dan juga kita ada sequence amino acids. Right? This is the translation of DNA sequence into its corresponding amino acid sequence. Okay. So note each has made up of 3 consecutive nucleotide. Okay. So now you have to do how many codons are there and how many aminos acid are there. Okay. So sekarang ni awak kena point what are the amino acid at this position. Okay. So dekat sini awak kena check dekat dalam window yang baru tu. Okay. Nucleotide number 10, what's amino acid? B, nucleotide number 85, nucleotide 218 and nucleotide 431. What is the name of amino acids presence? Okay. So now we will change or mutate the part of DNA sequence and see how it affect the proteins. Okay. Do the step number 22 to 27 below. Alright. Note down what happened to the protein and state the types of mutation it represents. Okay. So 18 in editor window, select nucleotide number 36 and change it into A. Okay, so awak cari dekat dalam tu, bukan yang dalam apa yang translate sekarang ni dekat dalam belakang tu pula. So tutup awak punya translate, buka balik awak punya editor window. Editor window ni adalah kita punya yang ada sequence tadi tu. Okay, editor window adalah kalau awak tengok dalam gambar rajah ni, bukan yang window depan ni, tapi awak tutup window depan ni, lepas awak check yang belakang ni. Okay. Right? Okay. So now awak tukar nucleotide nombor 36. And then translate back this new sequence tadi. Okay, translate awak pakai icon translate tadi alright. Say what types of mutation you create and what are the consequences of these mutations. Alright. And now select edit and undo typing to restore the original sequence. Okay, so tukar balik, lepas tu awak tengok balik. Sekarang ni kita kena change nucleotide number 68 to A pula. So report, what are types of mutation have created and what are the consequences of this mutation, alright? So print screen awak punya result tadi. And then kita tukar balik pula. Kita restore balik, ubah balik pada sequence asal dan kita tukar pada nucleotide nombor 13, we put A. And then print screen dan jawab soalan. Right? Okay, untuk 21, restore the sequence back, delete the G at the position number 61. Okay, so delete G ni bergantung kalau awak buka position number 61 tetapi bukan G, buang aja kita punya nucleus bases tu. Right? So tak semestinya apa G. G ni contoh kalau awak pakai apa strand yang saya cadangkan dalam, yang kita cadangkan dalam protokol ni. Okay, kalau awak pilih spesies lain, awak potong je. Alright. Sama same goes to number 22. Tapi position sama right? And go for the number 23, restore the sequence back and now select nucleotide from the number 121 to 140. Delete the entire of sequence of selection. Okay, highlight daripada nombor 121, 140, lepas tu awak buang. Then make a few random change and save the sequence under your names. Okay, so tadi tu awak dah buang awak punya sequence tu, sequence daripada original, lepas tu awak save as sequence yang baru tu under your names, and then we going to do the second process, right? So we will now learn how to align to sequence original dengan sequence yang kita deletekan tadi. Alright. The feature is useful when comparing close related genes. Okay, so now we're going to align to sequence, right? So open a new editor window. Okay, load back species original awak tadi tu. Okay, so window yang awak tadi yang awak potong, keluarkan balik yang baru So keluarkan yang apa original spesies yang asal tu, lepas tu yang awak telah delete tadi. So ada dua editor window yang awak keluarkan. So now click on align sequence icon. Okay, bila awak klik situ, there will be align sequence dialog will be open. And then make sure both sequence are selected for alignment and click OK. Right, apa yang awak dapat, awak akan buka dua ni, right? So result ini, make sure awak punya editor window ataupun API software awak mesti ada dua dibuka dekat situ. Bila awak buka, barulah ada dua nama ni. Kalau awak buka satu, dia akan jadi satu je. Alright, okay. So I will show you. Saya akan tunjuk macam mana cara dia. Okay. Alright. Okay. So make sure awak buka dua window editor tu. So awak buka align icon this one align sequence, and then you will get this display. Alright. And then click OK. A new window will open and display alignment of both sequence to each other. Okay. The sequence align function compare both sequence and try to match similar stretch of nucleotide. And then you will get the red area indicate sequence they are not mismatched. Okay. So akan dapat gambar rajah, dua gambar rajah ni, okay, A and B. Okay, A adalah kita punya sequence yang align dengan dua species, dua editor window tadi. Alright. So let's say yang atas ni adalah kita punya original, yang bawah tu adalah kita edit tadi. Okay. Right. And then the B is a other samples. Okay, this one nampak ni. So make sure bila kita buat tu dia akan keluar warna merah lah. So bila warna merah maknanya awak dapat lah yang mismatch dari situ. Right. Okay. Okay, that's all for today. Okay. Is there any question? Okay, is there any question sebelum saya bagi awak masa untuk buat benda ni? Okay. Alright, ada question lagi? So mohon semua faham Okay, kalau tak faham, ongoing buat kerja ni saya ada sampai pukul 5 So you can reach me on the WhatsApp ataupun any medium. Alright. So please bear in mind saya dah record video, saya dah record kita punya briefing ni. So nanti saya akan uploadkan dalam Google Classroom. So awak boleh download dan check balik apa yang saya beritahu tadi. Right. Oh, untuk PCR saya tak buat lagi. PCR no video alright. Okay, untuk strand tu, saya kata tadi strand awak boleh pilih semua jenis bakteria. Buka SCBI dan terus check bakteria apa Dengan syarat awak kena check the sequence is between 1 million maximum or minimum 15,000. Okay. So untuk awak nak check strand dekat mana, awak check balik video ni dekat mana awak nak tahu nombor dia. Nombor tu bilangan dia ada dekat bawah dekat tulis tu base pair. Bakteria boleh pilih bakteria apa pun. Jangan pakai bakteria yang dicadangkan dalam protokol ni. Okay. Okay, no infographic for this. Okay, tak ada infografik awak just gunakan Microsoft Word untuk buat kerja ni. Semua info-info gambar awak just masukkan dalam, awak print screen dan letakkan dalam Word. Boleh, right? Okay, saya akan tunjukkan cara nak buat, saya akan beritahu material yang boleh membantu awak dalam. Okay, boleh. Ikut format lab report. Okay. Okay, saya akan tunjukkan apa yang saya bagi awak dalam island alright. Okay, I will giving you this guideline works for special semester ni, alright. So apa yang dekat ni adalah material yang pertama, a lab manual MIC271 as a guideline, okay. And I will provide you several videos to help you for your doing these practicals, okay. A video that will guide you what is APA software all about and how the works process, how the works will done. Okay, saya akan bagi awak ada lebih kurang 5 laman web YouTube yang awak boleh tengok cara penggunaan API software. Alright. For this practical, just send to me full soft copy in the Microsoft Word. Okay, bentuk report dalam bentuk report. Okay. Right, is there any question yang boleh saya bantu lagi? So ada apa-apa soalan? Sebelum saya lepaskan awak untuk play around with the software. Is there any question? Okay, ada soalan lain? So untuk eksperimen saya ulang, tak perlu buat infographics. This one just tulis awak punya, buat macam manual lab biasa dan print screen semua result yang awak ada daripada software tersebut. Okay, so ada soalan?